And okay, so Simon will hopefully pop in here. But thank you all for coming. This is another thing in the series, and it's relatively simple. But we just, want to make these videos not only for you, but for other people that will eventually discover these tools. One of the things, at least on GNPS as well as a wider kind of metabolomics community, is there's new features, there's new links that show up, there's new integration, and we're all working together. And then eventually we think people will find them and click on them and ask, wait, what is this? So I just want to go over kind of what we're doing today, high level ideas, and then how we've integrated this tool into several related resources, and hopefully it'll help. So broadly, one of the problems that Simon actually came up with his first idea a year ago, and he's here somewhere. Wait, you can. So Simon Rogers, he's in Glasgow, and so I've known him for several years. But we were at the Metabolomics Society meeting in the Netherlands last summer, and he came to me and he was like, "Let's build this thing." He had this problem of not being able to get a good picture of a spectrum, nor the actual peaks of a spectrum automatically from a computer. So it's both a, kind of from a mass spectrometrist perspective, as well as a kind of a computer scientist perspective. And so I can appreciate both of those things. It's goddamn annoying to draw a spectrum and visualize it from several resources, not just a single one. And so the idea we had was, can we make a way that we can visualize and grab the data for a spectrum from any of these metabolomics online resources with a single click, and you never have to think about it again. So this piggybacks off the work of the proteomics group, where they've defined what they call a universal spectrum identifier, which helps you. It's like a URL for a spectrum, right? So you just have this long string that describes, what data set, what library, and things like that. That's automatically generated by a particular resource, and then some software can recognize that, get the data, and give it back to you. And so you can think about just carrying this identifier with you wherever you go. You put it in a manuscript, you put it in an analysis, and so you can always find the data. And so we built this nice web interface to do it. But additionally, what we did, this is where Wope comes along, is we had a really crappy way of drawing the spectrum. So Wope put out this really nice package, this Python package that can take the spectra and draw it and customize it in a variety of different formats, like PNG, SVG, as well as being interactive. And so kind of putting all these things together and then integrating all these resources, we got something. So we hope that it's just easy and you never have to think about it. And that's it. It's not, some of the other workshops that we've done were a little more complicated thinking about parameters, but it should be as simple as clicking a button and downloading an image and putting in your manuscript. And so broadly, one of the things we always found missing was automatically making nice looking vector graphics for editing in Illustrator and then putting into a manuscript. But then there's also the side consequence of, if you know this identifier, you can start linking back. And so there's you have QR codes and it's all fancy. So if you all know how to interact with them, try it out. But anyway, let me get this thing started. I've rambled long enough, but actually first, let's just quickly do introductions. Wope, do you want to introduce yourself?
Sure. Yeah. Thanks for the brilliant introduction of the USI project, Ming. I am Wout. I'm a postdoc in Peter Dollestein's lab at UCSD, working on a bunch of computational stuff, including this project, that this service that we'll introduce to you now. Like Ming said, it's just something simple, but it has made our life so much easier over the past few months, and we hope that it will make your lives a little bit easier as well.
And Simon, did you want to introduce yourself as well?
Yeah, I'm Simon Rogers, University of Glasgow, the computer science department. yeah, so Ming and I made the prototype for this sitting, drinking coffee when we should have been at talks at the Metabolomics meeting. Yeah, about a year ago. I'm actually not going to be here very long because this is the dining room of our house and it's dinner time now. So I'll disappear at some point, but nice to see you all.
Thanks, Simon. Yeah, I mean, honestly, I mean, talks are great, but the real action happens at the coffee hour, right? But it's seriously, as Wout said, it's made our lives a whole lot easier. And it's simple. It's just one of those things you're like, why does it should just work? So that's hopefully what we're going for. But as well as, so the core team is the three of us. And so one thing we are aware of is we are computer scientists. We think in that kind of style and automation, and you know, kind of a side excursion for us is mass spectrometry. So for you true mass spectrometrists that actually look at data and think about how to represent it in manuscripts and convey that knowledge, let us know what we're screwing up. There was one thing that we just saw, we didn't italicize M over Z. So it's a convention thing that we missed that we particularly don't think is useful, but you just have to let us know and we'll make things better. So to get started, let me share my screen. Is it this one? And so the goals of this particular workshop are, how can we start visualizing and customizing the spectra, just to give you a thing, and exploring where can we actually view spectra from. So we have some software that understands how to pull data from various resources, but we might be missing some. So we'll tell you what we can cover right now. And for those of you who are more well-versed in other resources that we're not covering, let us know what we're missing. And also, if you want to check it out, it's available online at metabolomics-usi.ucsd.edu. So this is all on the slides. You can download them and click on these links. So broadly, there's the high-level workflow that you probably had to do was, if you want to visualize a publication or just to kind of explore the data, is you have this LCMS file. So this is mostly LCMS data, though we do support GCMS now. But you have this LCMS file that hopefully is open format, like MZML or something like that. And you're like, oh, I want to visualize this spectrum in it, whether by scan number or retention time. So you have some process where you extract the MSMS, and then you get some intensity vector, and then you must take it to some piece of software and visualize it. A lot of vendor software does this automatically within it. But if you're doing it, you know, kind of vendor agnostic, this is kind of the broad outline. And so what I traditionally did in the past is I actually took this table in the middle and I went to Illustrator and I drew a bar graph. Not the best, it's so much work. It does look nice, but it's so much work. But both of these steps, you want to make it easy, right? Instead, what you can think about is, instead of the raw MZML file, if you're doing your analysis at any of these web services, whether GNPS, Mass Bank, or MSLDA, you should be able to extract it directly from there, which might have an underlying full LCMS run, but you've uploaded your data there, you've done some analysis, you should be able to extract and visualize the spectrum very easily. And so that's kind of the goal here. And so just kind of giving you an idea of what we can handle is, if you've also made your data public, your data set's public, you can extract directly from the raw data as well. And so for you programmers out there, if you want to do the extraction, you can extract it in JSON, you can extract it as a CSV, all through web APIs or through the user interface. So if you want to work with the data in that fashion. And additionally, you're able to visualize a spectrum automatically in a vector. You can resize it, you can customize it. And since we generate these images on the fly, what you can start doing is you can start embedding these images in other websites. So that's some of the things that we've done to make things easier. If you use GitHub and write Markdown, for those of you familiar with that, super easy to do. One thing, it's so easy. We started embedding like 500 spectra in one web page, and that kind of, it like really kills the server, that's our problem. So if you want to do that, you want to visualize everything at once without even thinking, go for it. And then we'll solve the technical challenges there. But so if you go to the actual web page of Metabolomics USI, the UCSC.edu, what you can see is you're agreed with this home page. And so you can enter these spectrum identifiers here and it'll resolve them. And so we'll address how do you create these later. Generally, you probably won't have to. But we also have example identifiers from multiple different resources, such as MSLDA, MotifDB from MSLDA, GNPS library spectra, a GNPS analysis, mass bank, if it's a public data set that's deposited in Massive, Metabolomics Workbench, metabolites. So those are for visualizing single spectra, but what we can also start supporting is doing mirror matches. So if you have two spectra that you want to view on top of each other, we support that as well. And so if you go ahead, so we'll do it somewhat interactive. If you go to metabolomics.ucsd.edu, and you go ahead and click on an example, any example, except the mirror spectrum one. So if you go ahead and do that, what it will do is it will take you to a page like this. And so there's several sections to think about. There's this top section here where you can actually get the exports. There's this QR code that you can start embedding in your papers or posters. So one of the things that was kind of cool is if you're at a poster, none of us are really at posters now, but if you put this on your poster and there's a spectrum, people can look at it on the phone and it'll be brought to this page. So it's one nice way to get people to the data so they can play with it instead of just kind of a printed image. As well as in the bottom here, you can start customizing the visualizations. And we'll in a second kind of live navigate you to this page and show you all the things you can start customizing. By no means is it exhaustive. Our kind of goal here was to help you customize to 90% of the most common things you'll do. That last 10%, it's a lot of complexity for us to put on the page. So you can bring the graphic to Illustrator and you can edit it there because it is a vector. So it's just kind of marrying the benefits of both. So we'll get you most of what you need, but for the final bits, you can edit it yourself. So that's the page. So, but now the question is, how do we get to this page? How do we get those particular identifiers? So I'll show you just kind of examples from different resources. And the first one is Mass Bank. So we worked together with the Mass Bank team. So Tobias, Stefan Neumann, Emma Szymanski, and Renee, I forget the last name, but that group. And they were awesome, and they put the link for the Mass Bank spectra directly on the Mass Bank accession page. So if you click this link, it'll automatically create the USI and take you to the right location. So for example, it'll take you to a location like this. So that's Mass Bank. Additionally, if you're at GNPS and you're on the library page, we also have a Metabolomics Spectrum Resolver link right here so that you can click on. It automatically formats it, automatically links out. You don't have to think about it. So if you want to embed library spectra in your manuscript, definitely the way to go. Additionally, if you are doing an analysis like molecular networking at GNPS, and this is an example for library search, what you can do is there's these buttons that show up now called USI links. So if you click that, it'll give you several options. If you're doing library search, it will give you a mirror match USI, so it'll start showing your spectrum versus the library, as well as you can view just your spectrum or just a library separately. So it makes it relatively straightforward to generate that. And then finally, from MS to LDA MotifDB, if you do, and also if you just do ms-lda.org, the analysis there, it'll show it as well. But there's links now that say Resolver MZ spec. So this link down here. So now all these different resources link to the same location. So that's all I wanted to show you regarding slides. So again, very, very straightforward. But now we'll take you through, we'll take you through a quick demo of the web interface, just to show what you can customize and things that you can actually download and interact with. And also we have a GitHub, so all the code is open source. And on the GitHub page is a README, and we're working on some better documentation, but it tells you the structure of the USI. And one particular word of caution is, as this is an unpublished piece of work, we are still iterating on some of the USI definitions. So how do you define these strings? And so we're undergoing one quick revision over the next couple weeks. And so the identifiers will change slightly. So don't put these in papers just yet. You can put the images in the papers, totally fine, but don't put the USIs just yet until the paper is actually published. What we're doing is we are coordinating with the proteomics folks to make sure that we're all happy about what the semantics are, and we're aligned there. But at least in terms of drawing spectra and making that available for publication, totally good to go. But anyway, I'll kick it over to Wout, and he'll show you what's going on there.
Thanks, Ming. I'll give a quick demo of the functionality of the website. It's largely gonna overlap with what Ming just showed during the slides. But if you go to the URL for our resolver, so Metabolomics usi.ucsd.edu, you get to the homepage. You can enter custom USIs here for to visualize a single spectrum, or two of them here to visualize a mirror plot. But for simplicity, let's just quickly use one of the examples here. So you see here we have the spectrum of a mass bank, entry and mass bank. Initially, it's visualized with some of our default settings. So we try to annotate the most intense peaks in small MZ windows. Note that actually indeed here, precursor MZ, the MZ here in the title isn't italicized, but it actually is in the x-axis label italics already. So there I actually thought about it. And like Ming said, there are a few customization options. Here a little bit lower, below the mirror plot. You can change the figure dimensions. You can change the mass range. So if you want to zoom in on a particular mass range, you can do that here. So let's, for example, look at these peaks here. So we zoom in on mass range 220 to 260 MZ. As always, update here below to make those changes, and then you get this kind of zoomed in region of your plot. Can also put a maximum intensity cutoff. So here, 20%, we're gonna look at this peak in a little bit more detail. Zoomed in here. Also, let's just zoom out again. Get the full plot. You can toggle the grid on and off. Here is something that's all pretty straightforward, right? Something that's kind of interesting is here. You have a list of all the peaks in the spectrum with their MZ value in italics and their intensity, and some checkboxes on the left that signify whether the peaks are annotated here in the spectrum or not. Like I mentioned, we use some kind of heuristics, some rules to figure out whether peaks should be annotated or not, but these might not always include the peaks that you're most interested in. So you can manually here select which peaks you want to annotate, and you can order it by intensity, this table, or by mass. Select and unselect here the peaks that you want to annotate. If you update again, you see that the corresponding peaks now have been annotated. And you can also control the precision of these MZ values in the annotations and whether they are rotated or not, like this. So some customization options. Like Ming said, this is not meant to be exhaustive because there's a lot of things you can do. Of course, you might need to do some final fine-tuning in Illustrator or something like that. But if you have some requests, some functionality that you think is missing, by all means, let us know. And then finally, besides these customization options, here are 4 download buttons. As Ming mentioned, the first 2 download the peak values in a few different formats. So either as JSON or as CSV, so that you can then process them yourself in your own, in some scripts. These 2 download the spectrum as a vector graphic, an SVG, or as a high resolution PNG. And that includes the customization changes that you've made here in this in the figure that you will download. And also here, you will see at the top of the page, you see the actual USI, its format. So if you are unsure how to construct one of these USIs, this is you can start from these and use them as an example. But also here you can see this is the link out to MassBank. All the information here you can see on MassBank. And then as Ming mentioned, MassBank and other resources have these links back to the USI. So you can easily go back and forth between these different resources. So this is for a single spectrum. You can also visualize mirror plots. Same, so we have 2 USIs on a greater mirror plot. Oops. What is this?
Wait, we'll just do the example.
Yes, I might have made a mistake in copy pasting that spectrum. Anyway, let's do the example. It's basically the same, right? In the mirror plot, the first one is the top spectrum, the second one is the bottom spectrum. You have this information in the header again, the peaks, the same drawing controls. Now you have 2 tables with peaks to annotate, one for the top spectrum, one for the bottom spectrum. The rest just stays exactly the same. Again, you can find the information for the input spectra here. You can go to their original resources and so on. Maybe like one last remark here, the cosine similarity that's computed here. Yeah, here it's perfect cosine because we've just used the same spectrum for this mirror plot. If it's a different spectrum, that's not going to be the same. Let's see. So example here. So we have 2 different spectra with their cosine similarity. Maybe one quick remark, this cosine similarity might not always match values you see in other resources such as GNPS, for example. Why is that? It's because there are different ways to compute this cosine score. Here, at the moment on the USI website, we only check for directly matching peaks, whereas on GNPS, the cosine similarity includes shifted peaks as well. So that's kind of more like a full spectrum alignment. So there might be some differences in the way the cosine similarities are computed between different resources. So keep that in mind. All right. It's like a brief introduction, like Ming said. It's pretty simple, but I mean, we like it. It has helped us in quickly visualizing stuff, sharing plots with other people, including them in publications and so on. And I hope you guys like it too. Yeah, we have some time for questions. If you have questions, suggestions, let us know.
Thanks, Wout. Yes, so there's one question I think from Chogwo. Sorry about that. But basically, it's about plotting from the raw spectra, raw data. So instead of a library spectrum. So let me just show you something really quick to just highlight. So the examples will show you it as well. So what we have here, I clicked on, just an example, the massive GNPS repository spectrum. And so this basically takes data from from an actual full LCMS run. And so from the identifier, it's actually a public data set and then a file name and then a scan number. So this is going in, reaching inside the MZML file and reaching out and taking out a single scan. So if you know what you want to plot and the data is public, it's very easy to construct one of these manually. Or if you've done it in GNPS, the links are already created automatically for you. And so it doesn't have to be a library spectrum, but this is a full LCMS run, a spectrum out of a full LCMS run.
Yeah. Another question that was asked is, what are the gray peaks in the mirror plot? So indeed, that goes back to what I mentioned about cosine similarity. So the peaks that are matched across the two spectra and that contribute to the cosine similarity, they are colored black for the top spectrum or green for the bottom spectrum. And the gray peaks are the ones that didn't match across both spectra. And to clarify how the cosine is calculated, so it's not actually bit vectors. It's no vector space or something like that. It's just like a peak to peak calculation. So we loop through both spectra. We check whether peaks in the top spectrum and the bottom spectrum can match to each other if they have similar mass. So currently we use a 0.02 MZ fragment mass tolerance. If they match to each other, we add those peaks as a potential match to a list. And then finally, we will, to avoid multiple peaks matching to avoid one peak in one spectrum matching to multiple peaks in the other spectrum, we then do a greedy selection of the best peaks that match to each other based on the highest intensities. So I hope that clarifies the way we do it, Charlie.
So there's a question about a local file. So the local files, yeah, so Woat's tool actually that plots these things, that's a Python package that you can use yourself. If it's a local file, then you'll have to write some code that interacts locally on your computer. So this only works because it knows how to retrieve the data from web resources. So if it's locally on your computer, that's not on the internet, right? So there's no way that it can actually retrieve it. So yeah, you just have to write your own kind of code to extract a spectrum and visualize it. But it is relatively doable. Yeah, any other questions? maybe one of the things we should think about, Wout, is kind of along with Krista's question, is you might not do molecular networking. You just want to visualize a bunch of the spectra, right? And so what if, okay, so this is what I'm thinking is, we take your spectra, you upload it to GNPS or some equivalent web resource. There is a workflow that literally all it does is it catalogs every single scan number in those files that you select. And it generates the USI for that. So it does literally no other processing. And then so it'll just create a set of links that you can click on based on scan number or spectrum, some spectrum identifier, and then you can create the visualization. It's the most simple thing, but it removes you having to understand, okay, what is the spectrum identifier, making sure the your data is available on some web resource. Simply, you just need to upload it, which we hope you're doing anyway. And then you don't have to think about, oh, I need to have my molecular networking settings right to make sure that the spectra actually shows up. The spectra will definitely show up. I don't know if that'll help. So it's a thought, it's an idea.
Indeed. Yes, indeed. We wouldn't need all the scans, but if we like output a list with links to USIs, then you can just like click on the ones that you need, select those, extract those.
Yeah, something super simple, yeah. And so then it just gets around the fact that we need to ask you for any information. You can just kind of post-process it. So anyway, it's an idea. Might not be a good idea. It is an idea. But anyway, that's really all we had to show you. It's nothing fancy. I don't know. We think hopefully it helps. Give us your thoughts if it's helpful or not, or where we can improve. One of the big things that we're doing is we're gonna redo like the cosine scoring. We're probably gonna change, as well as customized titles we're thinking about. And obviously the change in USI formats, but those are kind of, yeah, those are slated for the next release. But then there's some other things that we're being a little bit more adventurous on about, for those of you in the know, like splash hashes for spectra and kind of connecting everything together. But that's a separate thing. We're not gonna talk about it here. Yeah, so just giving you an idea, some of the uses of this that we've been using internally is if we want people to manually inspect spectra, to say for identifications or just giving people easy access. So one of the, this has been really nice. And so one of the things that I've done recently is there was from UIC, University of Illinois, Chicago, they put out a lockdown challenge. So we're, everybody's at home to solve some MSMS spectra to the structure. So this is kind of like CASME. It's just kind of a fun thing. It's kind of a challenge for people. But they provided their spectra, but they didn't provide the visualization. It's just a list of peaks. So we reformatted, made a data set. And then what we did was made a small Markdown page. So let me show that really quickly, just to kind of tease your imagination on some of the things you can do with this. But then we allowed people, so let me just paste it here. It visualized the spectra like super easily. And let me just share my screen for those of you who don't have access to it. But writing Markdown is super straightforward for those of you who haven't done it. And you can just embed these URL links for the spectra. So it automatically renders all of these in one shot. And so it makes it easy for you to inspect anything that you want. And then as Wout had said, you can customize the visualizations, right? And so here we particularly didn't, but what you could think about, especially on the MS1, is you can change the URL such that it bounds the lower and upper bound of the MZ, and so it zooms in on your peak of interest. And so, depending on your data, you can set some of these visualization presets, and then it just kind of works. And so just to give you an example of that, so if we go to this particular page, so there's an MS1. So let's do 470, and we update it. So now this zooms in. Okay, that's kind of ugly. This zooms in here. So we have the MS1, and so now if we copy this link image, you can just paste this wherever you want on any website, and it has all this information about the rendering. So if you specifically care about the rendering details in customizing and how you want to embed it, you can change that URL, and it'll make it a lot easier. So that's some of the ways that we've been playing around and making spectra visualization more accessible in like these areas that we didn't think about before. So not just manuscripts, but you can think about making really quick dashboards of a data set that you can share with collaborators as Markdown documents, and saying, look, click on, or if you're writing a manuscript, put the USI link directly in there. And so for you grad students out there that want your PI to look at the actual spectra, it's like, here's the link, and here's the figure, and here's the link if you want to kind of check each other's work. So these are some of the things that we think it can help with, but overall it's kind of in the same ethos. On the GNPS side, you can share an analysis URL, so you have full provenance. Here you can share the spectrum URL, so then everybody can go back and re-inspect it. So anyway, that's that's what we wanted to show you all. We'll go ahead and post the video, like 30 minutes video, after we're done. And we have a preprint, so if you want to read that, there's really not much guts in it. It's honestly like, here, it does things. But if you do use it, we would appreciate if you did cite it. But this is kind of a small thing that we hope is just, it's infrastructure. At GNPS, every week's infrastructure week. So it helps us and hopefully the rest of the community be more productive. So anyway, thoughts, questions, comments are fully appreciated. But anyway, thank you all for coming. You can join us at office hours. And next week, we will be doing, I believe, feature-based molecular networking again, but in Spanish. And so if you want to join for that, you are more than welcome to. If you do not speak Spanish like me, I will not be attending. But the following week, we will be doing other things that will be in relation to chemical directionality and looking at quants and metadata and things like that. So anyway, that's in 2 weeks, I guess. But we have things drawn out till late August or something like that. So hopefully we'll see you again. Thank you so much for coming.